All right. Welcome everyone to the second week of our ML for Protein Engineering seminar series. Today we have the honor of having Frances Arnold, Nobel laureate, advisor to the president, amongst many other things, to introduce our speaker. Go ahead, Frances.
Thanks, Kevin. And actually, I'd just like to thank you very much for organizing this seminar series. It was a great idea and excellent service to the community. So I'm grateful to be here to introduce Bruce Wittman, the second speaker in this great series. And Bruce just happens to be a member of my laboratory at Caltech. He completed his undergrad degree in chemistry and biochemistry at Washington University in St. Louis. And then after that, spent 2 years in the South San Francisco branch of Intrexon, where he helped develop their high throughput DNA construction pipeline, contributing automation, DNA sequencing, and software solutions. So he's doing his PhD in bioengineering at Caltech, where he's been developing machine learning tools to do a good job with engineering proteins. And in fact, he's a whiz at understanding both the experimental needs as well as the computational benefits, how computation benefits directed evolution. And that way he's been able to develop really practical workflows for protein engineering by evolution. So take it away, Bruce.
Thank you, Frances. Let me just pull up all my slides. All right. Okay, does everything look good? Great.
Yes, good.
All right. Thank you all for coming today. And today I'm going to be talking about kind of some general overview of machine learning assisted protein engineering using two strategies that we've developed in our group to highlight some of those challenges, something that we call focused training machine learning assisted directed evolution and every variant sequencing, or evSeq. This is a two-part talk, so it would actually be great if we could hold non-clarifying questions till the end, just to make sure we don't run way over time if, say, there are more questions on the first part than the second. All right, so let's just jump right into it and kind of talk about generally, what we're trying to do as protein engineers. And so obviously proteins are engineered from the sequence level, and it's typically beneficial to think about this, conceptualize it as this protein fitness landscape, where say we have our sequence represented here in the XY plane space mapped to some fitness, where fitness is some value that we care about. And protein engineering strategy that's going to be effective needs to be able to handle the challenges that come with navigating this protein fitness landscape. So for instance, problem one would be that sequence space is vast, 20 to the n, where n is the size of the protein that you're trying to engineer. And on top of that is largely devoid of function. Most proteins don't do anything. They don't do the thing that you want them to do. Problem two is that the relationship between sequence and fitness is very complex. We can't often rationalize what mutations will benefit the protein when we're engineering it. And problem three, and this just kind of compounds problems one and two, for many fitnesses of interest, we can't screen more than a few hundred or thousand candidates. One thing we can take advantage of, however, is that functional proteins tend to be clustered quite closely together. So if I blow up that little part of the cartoon landscape here, we'll see that oftentimes proteins are quite close together. Functional ones are all clustered. And this is something that directed evolution takes advantage of to evolve proteins. So most are probably familiar with directed evolution, but if you want to think about how it navigates this landscape, it's navigating it in kind of a greedy local search strategy. And so how that works, right? So we start with our parent gene over here. We're going to mutate, but we're only going to do a few mutations. We're not going to be making 20 plus mutations at once typically, and that's the local part of the search. We're going to evaluate for an improved protein, a protein with improved fitness, and we're going to fix the one that is best and send it into the next cycle. And that fixing the best and sending it into the next cycle, that's the greedy part of the search. So local few mutations, greedy, we're just taking the best. And so this solves all of those protein engineering challenges that I showed on the previous slide. Mainly, sequence space is vast and devoid of function, so we're going to use a local search. We're just going to focus on regions where we know there to be fit proteins. We don't understand the sequence fitness relationship, so we're going to use a greedy search. We're just going to take what's best and move forward, not really thinking about the underlying mechanism. Screening capacity is limited. Well, this is related to the first two, but we're going to use that local search to only look at regions, again, that have fit proteins. Now, while highly effective, this strategy does come with some disadvantages. Namely, in the ideal case, every cycle lasts 1 to 2 weeks, and many iterations are typically required to get an effective protein. Additionally, the reliance on a functional screen means that this is resource intensive. And what's something I'll come back to later, greedy searches can become trapped at local optima. To kind of overcome some of these disadvantages, machine learning has obviously come on the scene. And machine learning takes a related but different strategy to directed evolution. And what we're going to try and do with machine learning is actually map the protein fitness landscape. So for instance, we're going to select some training points shown here in these blue dots on our landscape. Using these training points, so these sequence fitness pairs, we're going to build a map of the landscape, so some approximate representation of our landscape. And then using this representation, we're going to use it to kind of search through the landscape and identify the top variants, so shown in green here at the tops of these peaks. This obviously also must solve some of those protein engineering challenges, and it does so as follows. Sequence space is vast, so we're going to use a model to navigate. It really increases our throughput. We don't need to go into the lab and screen for things. We can just do things in silico. We don't have a physical understanding of the sequence fitness relationship, but that's why we're going to learn from data. So obviously you can incorporate domain knowledge, and that will often improve models, but you don't have to get an efficient machine learning model. And again, screening capacity is limited, so we're just going to use a model, right? This effectively expands our ability to screen. But this comes with some disadvantages as well. And one of the main disadvantages is that we actually need sequence fitness data for training the most effective models. And what that means in turn is that we actually have to sequence all of the variants that we're screening. All of these training data points need to be sequenced so that we can generate those sequence fitness pairs. And also predicted designs must be constructed and evaluated in the lab, which can actually create some molecular biology challenges. So number two actually was covered at least in part by the last seminar series talk. But what I'm going to focus on today are actually these two. They're bolded here. You'll notice I put and devoid of function in parentheses here because it's something we often don't really think about when building these models. But we're also going to talk about how we can go about actually getting these sequences that we need for practical applications. So I'm going to focus on this one first and then switch over later to this at the end of the talk. But to talk about this problem where sequences are devoid of function, we're going to use this idea or procedure of focused training, machine learning assisted directed evolution, or ftMLDE. So to take a step back, earlier I mentioned one of the challenges of kind of what I'll refer to as traditional directed evolution is that with a greedy search, you can get trapped at local optima. So say, for instance, we have a landscape like this on the left. It's very lumpy. There are many local optima. And depending on where we start and the order in which we make mutations, we might end up at different optima. Obviously, we want to be at this one or this one, the global optimum. Again, global, whenever I'm talking about this, is global within some restricted region of sequence space. But you can also imagine, even if we do have only one peak, depending on the path that we take, it can take more iterations to arrive at that global optimum than maybe we want, right? There would be a shorter path in this landscape. But we have a trade-off because we're using this greedy search, this greedy local search, to minimize how much we need to how much we need to do in the lab to reach an optimized protein. And so just to kind of highlight this, kind of taking away that abstraction from the fitness landscape, let's imagine that we have 4 positions that we want to engineer at. So say we have this toy protein up here, those 4 positions that we might mutate are in red. And so we can represent our 4 positions as these 4 circles here. And if I were to do my local greedy walk and really try and minimize my screening burden, minimize in kind of the optimal case, the lowest possible screening burden, I would choose one position to perform saturation mutagenesis. So I test all 20 possible amino acids at that position, identify the best and fix it, and then repeat. And I'd repeat that for all 4 positions. Now you might imagine, depending on which position I choose to mutate first, or the order in which I make my mutations at these different positions, depending on the architecture of the landscape, I can arrive at very different locations. Actually, the only way to guarantee that I arrive at this global optimum would be to evaluate all possible combinations of mutations, so this simultaneous saturation mutagenesis, evaluate all possible combinations in parallel, so do an exhaustive search to find the best combinations. Obviously this, if I'm going around 4 times, screening 20 at each position, that is 80 variants in this example, but this is 20 to the 4th, so 160,000. So in most cases, this is not practical, which is why we rely on this, but we would like to be able to arrive at these more optimized proteins. And so this is where that idea of MLDE, machine learning assisted directed evolution, comes into play, where we're still going to do this simultaneous saturation mutagenesis, but rather than an exhaustive search, what we're going to do is we're just going to sample and characterize N variants. So go to the lab, get the sequences, get the fitnesses for these variants, and then train an ensemble of regressors to build that map of the fitness landscape, and then use that map to identify the top M predictions and test those. We could also fix the best and then move on to the next 4 positions. So the screening burdens in these cases, single step would be 80, exhaustive would be 160,000, but using MLDE, we can tune it N plus M. And typically we set this N, this M to keep us in the range of 10s to low hundreds of variants, basically to be competitive with this single step approach. But there's a problem when we try and sample and characterize N from a combinatorial library over here. And that's namely that combinatorial libraries are enriched in these kind of minimally or non-functional variants that we call holes. So imagine this is kind of the view of the landscape when we're doing our traditional directed evolution. We've really focused in on this local region that has our fit and functional variants. But if I want to do a combinatorial landscape, I've effectively expanded the search space. Now I am looking at more combinations that maybe I would never have accessed or even needed to look at doing that walk. And it's generally known that as more mutations are made to a protein, the probability that it retains function decreases exponentially. So this results in far more zeros in the landscape, in a combinatorial landscape, than say a single mutation landscape. What's more is that in practice, when we're looking at this, it's not this perfect flat plane of zeros out here. This is the real world. And so noise from screening instruments will add false signals to these true zeros. So our training data might be drawn from something that looks like this. What this means is that if I randomly sample training data from here, so if I randomly choose proteins from a fully combinatorial library, then I'm going to have training data that primarily consists of noisy, low fitness protein variants, which can be very problematic for training effective regression models used in, say, MLDE, both because of these false signals, but also because it provides minimal information about the extent to which different mutations impact protein fitness, only that certain combinations either completely eliminate it or allow fitness to be maintained. And so we can actually see this if we look at a real combinatorial protein fitness landscape shown here with GB1. So GB1 is this protein. It's that same toy protein that I showed earlier. And there is a data set out there that's the measurement of almost all possible combinations of mutations at these 4 positions. So about 150,000 out of 160,000 possible. Fitness in this case is defined as binding, how well this protein binds to an antibody. And because all these mutations are close together, we both have highly epistatic landscapes, so there are lots of local optima in this landscape. But we also have what I was talking about in the previous slide, most of the fitnesses where the maximum fitness is just normalized to 1, and that's what it'll be for the rest of this talk. Most of them are around 0, as we can see in this histogram. So most are around 0, and even if we zoom in over 100-fold, it's actually still quite difficult to see the very highest fitness variants. And so if I again randomly sample from this, most of my training data is 0. And we can show how this impacts machine learning assisted directed evolution performance through a simulation. So that's what I'll show on this slide. So because we have that full GB1 data set, we can actually simulate how MLDE would perform if, say, we were able to filter out some of those holes, some of those low fitness variants. And so we can go through the simulation conditions to actually show this. First thing we're going to do is we're going to remove the top 2/3 variants, or top 2/3 of those with the highest fitness from the GB1 data set. This is basically just to make sure that when we're gathering our training data, we're not cheating by gathering the very highest fitness variants from the beginning. And then from this bottom 1/3 of variance, we're going to sample 384 samples for training data. 50% will be below some threshold, so some threshold of fitness, and 50% will be above that threshold. And then we're going to evaluate the top 96 predictions on the full data set. So run the full MLDE procedure on this kind of artificially inflated training data with respect to fitness, and then see how the model behaves. We're going to look at the maximum fitness in these top 96 predictions as well as the mean. And so if I pull that up, we can do this simulation many hundreds of times. In fact, it's 2,000 total simulations per condition. So there are 5 conditions, so we have 10,000 total simulations represented in each of these plots. This top plot is the maximum fitness achieved in those top 96 predictions, and this bottom plot is the mean fitness achieved in those top 96 predictions. And what we can see, based on our control, where we're just sampling randomly, anything greater than or equal to 0, is that as we have this threshold, as we force the training data to be sampled from regions that are kind of more focused to regions that have high fitness variance, our performance improves. And things that are notable is that, even with our very weakest filter, so this 1%, that's 0.01 is the filter, the maximum fitness is 1, so it's around 1% of the maximum. If we can filter out fitness values with basically a 50% success rate, then that's enough to improve results. And what's more is that increasing this cutoff, so we've gone from around 1% with this orange line to around 8% with this green line. As we increase that cutoff, it does improve, as we would expect. We get better and better results. But the performance improvement, it's less than just going from no filter at all to having a filter. And so that suggests that even the weakest predictor, even the slightest ability to kind of guide us away from these holes can improve MLDE results. What I'll note is obviously in practice, we can't do this, right? We don't have the full data set. It's cheating to use these filters here to choose our training data based on known fitness labels. But there are strategies out there, and the number of strategies is growing quite a lot for doing what we call zero-shot prediction. So essentially prediction of protein fitness without actually going into the laboratory and gathering any data. And so you can use these to kind of get a sense of what proteins are fit, and then use that for guided collection of training data away from these holes. And so some examples of what might be used for this focused training data collection might be evolutionary conservation. Human knowledge, right? Some proteins have been very thoroughly studied, and we already know what mutations will either destroy or keep protein fitness. Transfer learning is a viable strategy. Generative modeling also works. Oftentimes this goes hand in hand with evolutionary conservation these days. Or rational modeling, for instance, prediction of the change in stability of a protein upon mutation. And so we can actually go through, and this is something I did for GB1, and test how well do each of these 3 work. In principle, these 2 should work as well, but these are the 3 that I looked at. So like I said, these 2 tend to go hand in hand, so I'll kind of group these for now. And zero-shot strategies based on sequence, well, they can either be local or globally focused. And so a local strategy would look at an MSA, a multiple sequence alignment, and essentially ask, with what frequency, and this is in the naive sense, with what frequency do I see a certain amino acid at this aligned position? And so if I see it more frequently, then it's more likely to be fit. Current models are far more complex than that, taking into account the different interactions between different positions, but that's the gist of sequence-based zero-shot prediction. We could also look at global strategies. And so this would be using, say, a model from protein language modeling, where you ask a model that's being trained to kind of fill in the blanks in a protein sequence. What is the probability of this amino acid being an alanine, cysteine, glycine, et cetera? And then ranking based on that probability that's returned. And so for GB1, I went and I tested strategies. The local strategies tested were EV mutation and deep sequence. And a number of strategies from the global side, anything from ESM, which is a very popular protein language modeling suite, PROTBERT and PROTBERT BFD, as well as the MSA Transformer. The MSA Transformer is kind of somewhere between local and global, doing this mass token prediction on MSAs rather than individual protein sequences. Without going into the details of how this is done, without unnecessary detail, we could show that this works for EV mutation in the MSA Transformer. At least it didn't work for deep sequence, and it didn't work for mask filling with the language models trained just on unaligned protein sequences. But what we have in these two plots on the x-axis, this is the rank returned by these zero-shot predictions from most likely to be fit to least likely to be fit, plotted against log fitness, log true fitness. And then this black line is a sliding median window size of 1,000. And really what we're looking for is kind of a tail up in over here, which indicates that we have, to some degree, the ability to predict fit variance. Obviously, it's not perfect. We have good variance out here in the lower ranks. And you'll note in EV mutation in particular, there are a lot of ties. That's that coarseness in this plot. And that's because there are very few alignable homologs to GB1. So we actually can't break the ties in many cases. The MSA Transformer can, even though it's trained on the same MSA, because it has this global information from its training as well. So this is a viable strategy in GB1 using either of these two. But we can also use rational modeling. And so if we predict, say, the change in stability of the protein upon mutation, we can get a similar plot. And it looks like this. So this works as well. And again, not perfect. We're missing some down here, but we have that uptick. We are able to enrich. And Triad is the tool that was used for this. It's a software suite for predicting the change in stability, among other things, of proteins upon mutation. All right, so the whole point of doing this, right, is to show that we can actually practically generate training data without knowing the full distribution of protein sequence ahead of time. And so we want to know, well, how does this do when we're doing our simulations, right? So we can return to that simulated MLDE. Now we're going to call it simulated focus training MLDE, because we're kind of guiding our training data towards these non-holey regions on the fitness landscapes. Once again, we're going to be doing 2,000 simulations per condition, but in this case, there are going to be far more conditions. We're going to be testing quite a bit more. So the first condition, we're going to be sampling 384 samples from the GB1 dataset for training data, all below some threshold in the zero-shot rank. So by threshold in this case, I mean the rank. So we'll sample from, say, the top 1,600 predicted variance according to zero-shot predictor, top 3,200, and so on. Effectively to test, how broadly can we search and still see any benefit. We're also going to look into different encoding strategies. I haven't really talked about this, but different representations of our proteins within the MLDE pipeline. So we're going to look at one hot as a baseline, a physicochemical, which will be referred to as Georgiev, and then learned encodings, which will be derived from the MSA Transformer. And again, as before, we're going to evaluate the top 96 predictions and compare the maximum fitness achieved to traditional and MLDE without that focus training. And so that's what popped up down here. On our y-axis here, this is the maximum fitness achieved in those top 96 predictions. Our gray here on its own, this is a traditional directed evolution simulation. So basically enumerating every possible path through the GB1 landscape from any variant that has non-zero fitness. Where do we end up? As we might expect on a landscape with many local optima, it's kind of, there's some lumps. This is where we kind of get trapped along the way. And then we have these major groupings over here. We have one hot. So this is the set of island plots. This is those generated from one-hot encoded sequences, the Georgiev and the MSA Transformer. This is all for EV mutation. So everything we're looking at in here was using EV mutation for focused training data. And what we have in these different groups now is gathering training data from those different thresholds. So like I said, gathering from the top 1600, the top 3200, and so on down to our pink random. There's a lot of information in here, but the key takeaway from this plot is that regardless of the threshold tested, so regardless of over here, we do better than both MLDE, always shown in pink, as well as traditional DE, again, in gray. And we can look at different zero-shot strategies. So rather than EV mutation, let's say we're looking at the MSA Transformer. This plot is exactly the same as the last one. The only difference is that we use the MSA Transformer for training data collection. And we see again that, regardless of our threshold tested, this focused training MLDE outcompetes both MLDE and traditional DE. And of course, we can look at triad as well. So this one would be using predicted change in stability to gather our training data. And this is where we see our most effective conditions overall. So actually, if we look at this point here, this is basically not a violin plot at this point. It's pretty much a line. The global maximum was achieved in 99.7% of the simulations. So we have 2,000 total simulations. So that's 6 that we didn't achieve the global optimum in this case. Compare that to this traditional strategy over here, where it achieves it around 1.2% of the time. So we're doing what we set out to do and achieving that global optimum more frequently using machine learning than we would with kind of that traditional greedy approach. So all that to say, for this section of the talk, using auxiliary information, so from sequence or structure, we can guide training data selection away from holes to enable more efficient MLDE. So that's kind of the first problem. How do we avoid these holes? How do we handle holes in fitness landscapes? But practically, we also need to be able to train these models. And normally we don't have these big data sets derived from things like deep mutational scanning available to us. We actually need to go into the lab and collect it. And so that brings us to that problem of how do we actually get sequence fitness data, the data that we need to train these models? Which brings me to this second strategy, every variant sequencing, or evSeq. And so just to highlight again the problem that we're looking at here, if we look at a traditional directed evolution strategy, so that's what I'm showing here. I'm kind of a little more general than that 4-circle one I showed earlier. If we look at this traditional strategy, we're going to generate diversity, we're going to measure fitness, we're going to keep the best, and we're going to repeat. But importantly, it actually doesn't matter what the sequences are in here, right? That's the whole point of using these directed evolution strategies, is that we can abstract away the fact that we don't understand sequence fitness relationships, so we don't actually need sequence. And so in this case, what we might do is just take the one that, say, had 5-fold improvement over parent and push it through to the next round of this cycle. If we're using kind of machine learning assisted protein engineering, then we don't have that luxury anymore. So the first part is the same. We're going to generate diversity. We're going to measure fitness. But now we also need the sequence, because we need the sequence paired to the fitness to model our data to predict improved variance to screen. And so even if we can maybe get away with sampling much less in this kind of machine learning assisted strategy, this is still quite expensive. And it's an added expense relative to the traditional pipeline. And so even if maybe we can achieve much higher fitness variance using machine learning, it doesn't really matter if the actual cost of running the experiment basically undoes that. Efficiency is made up of many components. So the most efficient machine learning assisted protein engineering strategy would require both the same total cost and effort for data collection as traditional approaches, which is a tall order considering that we don't actually need that sequence information. That's not to say, though, that we don't sequence in traditional directed evolution, and we do. Normally, what's done is just the very top variant or variants will be sequenced, not out of need, but more out of quality control, and it helps with the next molecular biology stages. And so normally what we would do, we would sequence just the top few variants using what is called Sanger sequencing. That's kind of the go-to strategy for traditional directed evolution. And the total cost of that, it'll vary from experiment to experiment, but I've kind of put it as a fairly conservative $20 on this plot. That's this dotted line down here. But Sanger sequencing, it scales linearly with the number of sequence variants. So we might be able to get away, with 5, 10 sequences using Sanger. But as we increase the number of sequence variants, obviously our total cost goes up. And so we can frame this just kind of tying back to MLDE. Say I wanted to generate, 6 plates of data for run with MLDE, that strategy I was talking about in the first part. We've done this using this in the lab, and it cost us about $1,700 to do this with Sanger sequencing, which is obviously quite expensive. Now, the reason that we use Sanger sequencing is typically because it's very accessible. You just need to send a sample off to some sequencing provider and they'll handle it all. You don't need to be familiar with sequencing. You just need to be able to isolate your DNA. But there are other strategies out there. Most, I'm sure, are familiar with next generation sequencing. But there's a key difference that we can take advantage of between next generation sequencing and Sanger. And this key difference, not worrying about all the other details, the key difference is in how the data is returned. So let's say we have our population of DNA here. We have two variants basically in this population. If I were to do this via Sanger, we're going to get something back that looks like this. It's going to be one read per sample. It's just going to be this chromatogram, and at the positions that are mutated relative to one another, it's not going to know what's what. It's just going to tell us mixed population, but we don't know exactly what those sequences were. NGS, on the other hand, will return millions of reads per sample. And these reads returned are kind of equivalent to a random draw from this population of DNA molecules. So what we'll see is we'll get some number of reads, some map to one portion of the population, and the others map to the other portion of the population. So the key high-level difference is that NGS can deconvolute mixed populations while Sanger cannot. And this has actually been taken advantage of for a long time to basically put multiple samples in the same Sanger run using a strategy known as molecular barcoding. So brief background on molecular barcoding. Say I have n number of samples that I want to sequence all together, right? I don't need those millions of reads that NGS returns. I want to sequence these all together. So what I'm going to do is I'm going to tag each of these samples with a known piece of DNA. So I might say quadruple A, well, that goes to sample 1, quadruple T, that goes to sample 2, and so on down to sample N. And so when I pool these and then I run NGS, I'm going to get all my reads back. And now I can use these barcodes to map the reads back to my original sample. And so the reason that this is valuable is because it enables what's called multiplexed NGS. And so we can actually combine multiple samples in the same run, multiplexing, and that can reduce the per sample cost. And that's what I'm showing here. So on the x-axis, number of sequence samples in both of these plots. And then on the y-axis, cost per sample in the left plot, and the number of reads per sample on the right plot. And so you'll see as the number of sequence samples goes up, our cost per sample goes down at the expense of the number of reads per sample. At 600 samples, it becomes more efficient using my cost estimates here to run multiplexed NGS on a MySeq than it does to use Sanger sequencing, the per sample cost. So it's great. We can use NGS and then we can kind of minimize the per sample cost. We can drop that cost. But notice the total cost up here for one sample, right? The cost of a single NGS run outsourcing will be around $2,000, which means that if we, yes, we can get per sample better, but we're still 100-fold more expensive than that $20 Sanger sequencing that I showed before. But notice also this line here, the minimum number of reads needed. It's actually much lower than what we get to, even at this 1,000 point where we're multiplexing 1,000 samples, it's much lower. And so this is where we kind of come to this idea of every variant sequencing, or the strategy that it uses to drop those reads even further. So we have more reads than we need out here, and we want to have a $20 total cost. So the total cost per sample when multiplexing 96 variants is $20. So on this slide, 96 is around here. We're looking at around $20 total. And the total number of reads that we would get would be around 250,000. So what if we just multiplex this again, right? What if we had another barcode, set of barcodes within this sequence? And so now we take this single sample in the multiplex run, we spread it even further. We spread the cost further, we spread the reads further. And that's what this plot here is showing. So we can, it's basically the same as before, right? The same trends that we're seeing, but the important difference is that now we are more cost effective than Sanger at 7 samples, and we still have more reads than needed for reliable sequencing, even at 1,000 total samples. All for this $20 cost. Now, I will highlight that this assumes that all variability occurs within the read length, because we're assuming that we can capture all of our variability in this sequence with this kind of one-to-one mapping. So that's the cost part handled. We can spread these reads further in NGS, and we can get a total cost of $20 and still sequence everything. But how about the effort part? Because that's important too. This needs to be cost effective and effort effective. So we designed this workflow to fit into the protein engineering pipeline, the existing protein engineering pipeline, using only PCR. That's all that's actually needed to run this, because the rest is outsourced. So I won't go heavily into the details here because the technical details don't really matter that much. But the full pipeline is you start from overnight cultures. You're going to do a PCR to append what we call the evSeq barcodes. These are inline barcodes. Just pool everything down because it's barcoded. We can pool everything. A quick purification followed by another pooling, and then it's outsourced. And from there on, we don't need to worry about anything that happens. But that's when the provider will add their other set of barcodes. So that's the 2-stage barcoding and how we're spreading reads even further. This will be run as a sample in multiplexed NGS. Reads will be returned, and then we have software that will map everything back and give sequences in these wells. I want to highlight a few parts in this, though, and why this kind of drops the effort. The first one being we're using overnight culture. So this is actually something that's produced during the typical protein engineering workflow. So this is our template DNA, actually, to go into the PCRs. We just throw in the culture directly rather than doing any purification. I'll note that there are 2 sets of primers in here. One is site-specific. So these have no barcodes on them. They just point to the region where we know the variability is going to be. And the other set is barcode. So the reason we do 2-stage is because this actually means that we only need 1 site-specific pair per position, but we can recycle the barcode primers. So essentially that drops the cost of ordering primers by a lot. Here's the key part, though, and that's really in the outsourcing. Because like I mentioned, one of the key things that's interesting for Sanger and why it's so widely used is because it can be completely outsourced. You don't need to understand how it works to use it. And that's what's happening here as well. So this sample is just being run with samples from other users, evSeq or otherwise, in some pipeline in a lab that is not yours, right? All you need to do is the PCR. This means that data is returned quickly because you don't need to fill up the full run, but it also offloads the effort. And then the final piece is this software, the mapping everything back. And it's important to note that, when using this needs to also be very easy to use. It needs to be accessible to everyone. So the software that we wrote for this, it's accessible to people with varying computational backgrounds. You can run it out of a GUI, for instance, but you can also run it interactive in Python in a Jupyter notebook or from a command line interface. But the data that it returns is also kind of designed for people with different levels of sequencing experience. So here are 2 example outputs. One is this kind of high-level plate map view. So this is just a plate that we sequenced using evSeq, where we have the identity of the mutant in each well identified, given back to us in this kind of high-level graphic. But also returned are these kind of gritty details of how the sequencing went for each well. How many reads did we get for each well that came back? What was the alignment frequency, right? How frequently did we see this identified variant in there? But the important thing that you can do is once you have this sequencing data is that there are also tools in this pipeline that let you build these maps, these comprehensive sequence fitness maps of libraries. So this isn't a plate map here, this is just a heat map. So we evaluated a number of different positions in a protein of interest. You can see based on the coloring, we're getting very different responses in our fitness. And now we know for each position, how does an alanine do? How does a cysteine do? And so on. And so just kind of summarize, where we were and where we are with evSeq. So where we were, we had this kind of pros and cons table, right? The pros are obviously data for machine learning assisted protein engineering, but there are also qualities for pros for people who aren't interested in machine learning at all. Mainly, you can evaluate your library quality, evaluate your screening quality, because this is going to give you replicates and information on how well your library was constructed. And of course, once you have these big maps of sequence fitness data, you can derive biochemical insight. But this was outweighed by the cons, namely, we don't need to do this for directed evolution to work, and sequencing costs time and money. So with these evSeq tools that we've developed, this is gone, right? I mean, we're never going to get rid of this. We're always going to not need sequencing for directed evolution, but we've balanced the amount of money and time it takes to run this, which has kind of flipped this inequality here. Now I'll highlight this because I've been talking about this from the perspective of people who are interested in machine learning and how this lets us use these tools that we've been developing. But it's also very important to note that this is useful for people who aren't interested in machine learning. And with widespread adoption, we can kind of tap into that data generation pipeline that is traditional directed evolution. Namely, you can kind of do back of the envelope, hundreds to thousands of variants will be evaluated in a directed evolution campaign. Thousands of groups perform directed evolution. And so there's the potential for then millions of labeled data points that we can collect and use for machine learning. So this brings me to the conclusions from both of these stories and where they converge. So from the ftMLDE side of things, we can see that elimination of holes in training data improves MLDE results. And we would expect that this generalizes to other systems as well. Holes aren't a unique problem to MLDE. This is a problem whenever you're trying to apply regression to a fitness landscape. There are many existing zero-shot strategies. I certainly didn't comprehensively review them in this talk that we can use to design focused training data collection to get fitness-enriched training data. As we saw, ftMLDE outperforms a greedy walk by 83-fold. It outperforms the MLDE, the standard version, by around 10-fold. And just if anybody's interested, all of that information is generated from scripts for reproducing this work on our GitHub. From the evSeq side, we have our other conclusions. How do we handle this lack of sequence fitness data or the inability to get sequence fitness data? And that's namely that we can now get sequence fitness information from targeted mutagenesis. Now, targeted is important because we are focusing all of these reads to targeted areas. And so this relies on people, of course, using things like site saturation mutagenesis or random mutagenesis to small tiles. But these are pretty common practices in directed evolution anyway. This obviously allows implementation of MLPE. It has the potential to create growth in databases that can support development of strategies based on supervised learning. And again, all of the software and extensive documentation is available on our GitHub. So I'll finish it here just with some acknowledgements. Obviously, thank you to the organizers for organizing this, Francis, for your support on these projects, as well as the nice introduction. Sabine, our lab manager, for keeping everything afloat and really talking through on these projects. These are pictures of the full group on our last group outing. Everybody in this group has been important for developing these tools because all of these are developed kind of with wet lab scientists in mind. So keeping those conversations going are very valuable. I also have to highlight Patrick in the back here and Kadina, who helped a lot with the development of evSeq. They're my collaborators on that project. And of course, thank you all for listening, and I will be happy to answer questions at this point.
All right, thank you, Bruce. So there is a number of questions in the chat that I'll hand to you. So first, from Will Steele, says, hi, Bruce. What approach for zero-shot learning would you take for proteins in which the region you are interested in engineering shows very little sequence conservation? For example, antibody complementarity determining regions?
Yeah, it's a great question. I mean, the zero-shot strategy you choose is going to make or break this works, and it will vary depending on the protein and the fitness that you're looking at. GB1 has very low homology, as you kind of saw through those coarse grain predictions of things like EV mutation. I don't know your system perfectly well, so I can't comment exactly. But things that are global determinants of protein fitness will kind of always be effective zero-shot strategies. So for instance, predicted change in protein stability would be one. Predicted change in protein conformation upon mutation, right? If the protein conformation changes wildly, then you can kind of have a good guess that it's not going to retain the same fitness. So kind of thinking through what things might impact your fitness is how I would approach it.
Thanks. So I have a suggestion that I'll let people unmute and ask their own questions. So Adam Damry, if you want to do that, or I can read it for you.
Sure. Hi, Bruce. Thanks for the talk. That was wonderful. I just had a question regarding your zero-shot learning with the basically computational protein design algorithm. The triad force field, if I remember correctly, was partially trained on GB1, and CPD energies are often not very correlative to real stability. So I was just wondering if you'd have any insight on the performance of that method for other less well-behaved proteins for that kind of algorithm.
Yeah, it's a great question. Yeah, I mean, it's being validated on a number of things that were held out as well. Triad, generally is quite robust at making those predictions. If GB1 is in there, it's obviously going to perform better on GB1 as well. As for how well it will work on different systems, again, there's no one size fits all answer to that problem because it's going to depend on a number of factors. When I looked at GB1, there were different things that determined how well Triad would behave, for instance. So with fixed backbone, it tended to perform fairly well as a zero-shot predictor. But once you introduce flexible backbone, it didn't work so well anymore. But if you took that flexible backbone and then ranked based on something like root mean squared deviation, right? So change in the conformation of the protein structure, then it started working well again. So it's a difficult question to answer. There isn't an obvious answer.
Thanks.
All right, next up is Miguel A. Gonzalez. All right, I'll read Miguel's question. Hi, Bruce. How much can the concept of whole be influenced by the high throughput assay used to measure the fitness values? I.e., are holes consistent with biological properties like expression, thermodynamic stability, catalytic activity, etc.?
Yeah, that's a good question. So the general rule of thumb that you could follow is that as you add more mutations to a protein, you're going to see more holes. But the rate at which you see more holes will depend on where those mutations are. So for instance, if you're making 4 mutations right in the active site of a protein, probably everything isn't going to work, right? Except for those few that remain close to the protein. That is probably a chemical thing in that case. But you can also imagine if you make 100 mutations to the surface of the protein, you're going to see a lot of holes there. And in that case, it's a stability thing. So what causes holes really depends on where the mutations are made and the mechanism by which fitness decreases. For higher throughput screens, I mean, holes are just, a function of the fitness landscape. So if you're gathering more training data randomly, the way that you can think about it is it's okay if you have a lot of training data to have these holes in your training data, right? You're just comprehensively mapping the protein fitness landscape then. The problem more arises when you're in kind of this low training data regime where you might be drawn off target by a hole, or maybe your model doesn't learn as much as it otherwise would because there's a hole in there. That's why we called it focus training, because really what you're doing is you're just focusing your training data collection efforts on the regions of the landscape that have the pieces that have the fitness that you're interested in, right? You want to more accurately model those high fitness regions and the low fitness regions.
Thanks, Bruce. And I have one in this first part of our talk too. So when you rank all the possible variants and you set a cutoff, right? You're using a hard cutoff and you evaluate a number of cutoffs in your test case.
Yep.
Would you comment on the possibility of using a soft cutoff or like an energy to give you a probability of selection for each variant?
Yeah, it's a great question. I haven't looked into it, but it's obviously a next step because a hard cutoff is extremely arbitrary. And that was one of the reasons that I tested so many effectively to see, how important is this cutoff. But yeah, a soft cutoff would be a sensible next step.
Yeah, but either way, how would you think about choosing like the temperature for the soft cutoff or the hard cutoff itself?
Yeah, again, it'll depend on the protein, right? So imagine that you have many holes, right? You expect that most of the landscape is dead. Then you're going to want to set some cutoff that's a little harsher, right? It's not going to be as forgiving versus if you have one where you expect everything to pretty much be functional still, then maybe you have an even softer cutoff. So again, it comes down to there is no one single answer. It's going to depend on the protein system. It would be great to kind of develop heuristics to figure that out. You could also maybe imagine some kind of meta learning if you had enough of these data sets to actually look into, right? What should these cutoffs be? That could be a strategy that you would take. But we don't have many of these data sets, which makes these cutoffs quite difficult to learn. We have n equals 1 right now.
Thanks. So next is Tian Yang.
Hi, Bruce. Hi, everyone. So it was a great talk, and I was actually pretty excited about the evSeq when I read the preprint. And I'm actually thinking about applying it to my own project, but I'm actually planning things out. One thing comes to my mind. It seems like you're still doing 2 rounds of PCR reaction for each variant. And
yes.
You have a library size that has like greater than like 1,000. If it's like $1 per variant, it's still going to be like $1,000.
Yeah, I didn't include those costs, but the PCR. So we use that PCR. We do a very small reaction volume for the PCR, and we use TAC. And when we do it in our lab, it's fractions of a cent per PCR. So yes, that cost definitely factors in, but the major cost is still the sequencing cost. We can definitely talk offline about how to do that if you're worried about the PCR cost.
Cool. Thank you.
Yeah.
Next up is Joshua's iPhone.
Hello, Bruce. Can you guys hear me?
Yep.
Hey, thanks for the talk, and also thanks for the paper, and thanks for the code on GitLab. It's really great to see this out. And right now we're implementing evSeq now in our lab, and the question I have is around the limitations of short read sequencing. So if you want to sequence the entire protein, like how do you approach that? At what step do you pull?
It's a great question. It's something we're looking into expanding, right? That would kind of be version 2. How do we tackle the full protein? So the strategies that we've been using in the lab to kind of handle the short reads are more of a tile strategy. So say you wanted to randomly mutate the full protein, you just restrict it to a tile rather than the full thing, right? You capture all that variability in a tile. If you want to do a full gene that's outside of the gene or outside of the read length, then right now what you're limited to, unfortunately, is just kind of piecing together different reads, right? So you would have evSeq you do a PCR of one fragment, then a PCR of the next fragment that overlaps with it, and so on, which kind of breaks down really beyond 2 or 3. It's more a limitation of the system right now that we're hoping to address in the future.
May I ask a follow-up question? And that is, have you tried the long read sequencing?
Yeah, that's absolutely. We're absolutely interested in looking into it. We have really just started looking at that stuff right now. We developed this around, obviously, Illumina Tech, but yeah, we are looking into MinION and things like that.
Okay, very cool. Thank you.
All right. Those are all the questions from the chat. Does anybody else have questions? If not, please join me in thanking Bruce and Francis. And hopefully I will see you all in 2 weeks.
Thanks, everyone.